Good morning, and thanks for taking a few minutes to hear a bit about Cabaletta. This is our disclaimer regarding forward-looking statements I may make today. So Cabaletta exists to develop and launch the first targeted curative cellular therapies for patients with autoimmune diseases. This slide has actually been with the company since the day we had our first corporate slides made in 2018. We're doing this by pursuing two parallel strategies. The CARTA strategy, with our lead program, Cabaletta 201, which is a 4-1BB containing CD19 CAR-T product, is being developed in three parallel clinical trials, one for lupus, one for myositis, and one for scleroderma. And in these trials, we're starting with what we believe to be a fully therapeutic initial dose, making these not dose-finding studies, but actually studies to explore the safety of Cabaletta 201 at the intended dose that we expect to use. And I'll go into that in some detail. In addition, when we think about Cabaletta 201 execution in our clinical program, each of these trials doesn't have one cohort. Each of them has 2 or 3 parallel cohorts to enroll every month or even faster than that. So when all 3 clinical studies are up and running, we have the potential to enroll 7 patients per month. Cabaletta 201 is exciting to us because it is specifically engineered for patients with autoimmune disease. We have a fully human CD19 binder, which allows for redosing if and when it's needed. We don't expect that it will be needed based on the academic data we've seen so far, but if it is needed, it'll be good to have. That fully human binder has been studied in about 20 patients in an oncology setting as part of a tandem CAR, a CD19, CD22 CAR. And it has been reported that in that study, this binder that we are using in Cabaletta 201 had an excellent safety profile that would be perfectly suitable for autoimmune patients. The 4-1BB costimulatory domain that we are using is widely acknowledged, particularly in the treatment of lymphoma, but also across all cancers, to be a lower risk profile than the alternative, the CD28 costimulatory domain. And so we've really designed our product to appear and to behave as similarly as possible to the academic product that has been used with such great fanfare over the past couple of years. The product has the potential to cure or at the very least stop active disease in its tracks across a host of autoimmune diseases. It's really exciting because it will reset the immune system, apparently. And in that context, we're starting to see more and more academic data come out. We'll talk about that. And some of the companies that are entering the field are starting to use investigator-initiated clinical studies in Europe, a very fast way to generate non-regulated data, but it's useful, and it's particularly useful for us because that data helps guide our decision-making, as it did with the data that Professor Schett, the academic in Germany who published the exciting studies that we've all seen, has done. So we're really thrilled with the progress that the industry is making. There's an endless number of patients that need treatment, and being at the forefront of that in the United States with these phase 1/2 clinical trials across what we believe to be the broadest program in the industry is really thrilling for our team. Our legacy platform strategy is the CAR-T strategy, which is designed to kill only the pathogenic autoreactive B cells in a patient with pemphigus vulgaris, for example, or with myasthenia gravis. And in those trials, we have seen that this product for pemphigus, which is designed to be very safe, is very safe, even at very high doses. But without preconditioning, we didn't see the efficacy we would have liked to see. So we're running now a trial with Flu-Cy preconditioning as well as IVIG in order to determine if we can generate the sort of efficacy that the animal models suggested might be present in those patients. As well, a myasthenia gravis program is up and running in that legacy platform. So just to give you a sense of the breadth, the scale, the scope of the opportunity, as if lupus with its 330,000 patients in the United States alone wouldn't be enough, as if myositis with 66,000 patients in the US with really no options wouldn't be enough, as if scleroderma wouldn't be enough. This list is quite valid from a biologic standpoint. It's hard to imagine that we're not going to see efficacy across most of these diseases at some point. Our pipeline, as shown here, we tend to take a pretty conservative view. We expect to start opening clinical sites very shortly for both scleroderma and then myositis. In due course, we'll begin to publish the opening of those sites and talk about the enrollment of patients, with the objective being to do what we said we would do a year ago, actually, to the day. We said we would have initial clinical data from CABA 201 patients in the first half of 2024, and I stand here today to tell you that I am more confident that we will hit that milestone than I've ever been, and to reiterate that over the last 5 years, we have never missed a clinical milestone. And I want you to think back through COVID what that really says about our company. Our people are committed to hitting our publicly stated milestones, and this is no exception. So let's talk a bit about that academic data. It's thrilling, right? I mean, the holy grail would be if you could reset the immune system. And lo and behold, we form a company because we think CART19 is not going to be safe enough for autoimmune patients. And it turns out the strategy we're using, as elegant as it was not the right horse to be riding for the broad impact that we sought to have as a company. And so we looked at the academic data, and here's what we saw. The emerging clinical data with a 4-1BB co-stim domain in a CART19 product in an academic setting, we see from 2 different investigators 8 lupus patients, lupus nephritis specifically, 4 myositis patients, and 4 systemic sclerosis or scleroderma patients treated. 100% of patients have either a complete response, elimination of total disease in the case of lupus and myositis, or in the case of scleroderma, what appears to be an elimination of active inflammatory, active disease progression. It's remarkable data. What I think makes it more remarkable is the safety profile. So we all stand here in cell therapy. We're very familiar with the idea of CRS and ICANS, but the number one side effect in these patients was fever. Like, let that sink in for a minute when the alternative is cyclophosphamide, azathioprine, rituximab, high-dose steroids, and all the things you need to manage the patient who's going to have side effects to all those drugs. The number one side effect to CART19 4-1BB co-stim domain therapy out of 15 patients reported in the literature so far is fever. 8 patients out of 15 had a fever. About half of them got Tylenol and they were fine. About the other half got a single dose of tocilizumab and they resolved. 1 patient had a grade 2 CRS. They came into the study with myositis short of breath. That was their baseline. When they developed a fever, they got more short of breath. They got a 2-liter nasal cannula. They got a dose of tocilizumab. They were fine, grade 2 CRS. So you had a total of about half, a little more than half of patients had CRS that seems to be completely manageable. In addition, there was 1 patient who had a ICANS, what was described as ICANS, a bout of dizziness on about day 8 or 10. That dizziness was resolved with a single dose of steroids. That's categorized as ICANS. At the end of the day, the B cells repopulated. So you have this effective, durable, up to 2 years of follow-up now published across all these indications. You have durable therapy, complete responses, or halting of active disease progression in the case of scleroderma, and a safe profile, and the return of a normal B-cell population between 3 and 7 months in every patient. It's really just, it's a dream come true for patients who are suffering way too long. And on there you saw the data sets and the footnotes that you should take a look at the articles and really get deep on it if you'd like, because it is as astonishing as it appears. So CABA 201 was designed to look just like that product. So what do I mean by that? In vitro and in vivo studies shown here demonstrate both published, peer-reviewed, and replicated by our scientists the comparable bioactivity of our fully human binder to the murine binder, the FMC63 binder that was used by Professor Shedd in his trials. The similar bioactivity and the binding of the same epitopes because of the design of CABA 201 with a 4-1BB co-stim domain. We argued, and frankly we didn't argue, we just put the statements into our IND filing for each of our studies. With FDA, we said, look, a dose-finding study makes no sense here. We know our starting dose. So unlike every other company that we're familiar with, we're not doing dose finding. We've moved to the next step. And for everybody who knows cell therapy, you know that dose finding is a painful and a long process. It's 1 patient per month, next dose after 3 patients, another 3, then you expand to 6 patients. We're expanding to 6 patients as our first step. So in every one of the cohorts that we're treating 6 patients. So let's talk about myositis. About 66,000 patients in 3 different subtypes of disease. Interstitial lung disease is a killer for these patients. The first patient that was treated, and I showed the data briefly in summary fashion, was walking 10 meters to the bathroom with 40% oxygen. About 6 months after therapy, they were reported to be walking 5 kilometers. Side effect profile, the CPK went up immediately after the therapy. It came back down to normal. The inflammation on MRI, gone. Remarkable in every dimension. So this is a very high unmet need. The only available therapy is IVIG. The cost, about $500,000 per year, and only indicated for the dermatomyositis subtype. Think about what a clinical study could look like when you're comparing your product to the best available standard of care. We're starting out, as I said, every trial is going to look the same, and for time I'm not going to go through each one in detail, but this one I'll post for you that says 3 subtypes of myositis. We treat patients, 6 patients in each cohort. When we get to the 6th patient, we're talking to FDA about a registrational trial. So that's our path, and there are currently 7 different paths, 7 different ways to get to that end game. This one happens to be particularly attractive because, number one, there is no other IND for myositis with CART19 in the United States. In contrast to lupus, there are a few. And there are no alternatives for these patients. There aren't like 6 therapies that you treat and you fail and This is a bad disease with no treatment. It's a perfect use of cellular therapy. Lupus and lupus nephritis. Lupus nephritis, you'll either be dead or have kidney failure, about 1 out of 4 patients over the next decade. So that's the prognosis, despite best standard of care. You take those patients, about 135,000 in the US. Earlier today we heard from a panel presentation that Gilead had treated a total with Yescarta of about 16,000 patients. Just like let that sink in for a second. 16,000 patients, 330,000 with lupus. There is a tremendous opportunity and room for everybody, but we're thrilled that we're in the position we are with the trial designs and the path, we hope, to a launch that will bring us first to market and allow us to have the broadest portfolio in the industry. The trial design here is exactly the same, 2 groups, lupus nephritis, which is what Professor Schett treated, and lupus, which hasn't been demonstrated to be a responder yet, but one could assume, given the pathology of the disease, that it will be the same. Nonetheless, we have to study it, and in case it's not, we have a separate cohort just focused on lupus nephritis. Systemic sclerosis. For those who are physicians, you'll be familiar with this. If you're not, just picture that you're being mummified. Your skin is becoming more and more stiff and fibrotic until you can't expand your lungs anymore, and oh, by the way, your lungs are becoming fibrotic too. That's the middle-aged woman who has systemic sclerosis, and in 10 years, an average lifespan of 12 years. These are the diseases we're treating. These are really serious unmet needs and compelling both for patients, for the physicians, for use of CAR-T therapy. It's a perfect place to enter into the market. And again, the clinical trial design, very similar. I'll note that the inclusion and exclusion criteria are very broad in our trials. We don't know if treating patients with less interstitial lung disease is going to be better than those with more. So we're doing 2 cohorts. We'll find out. Our people and our product and our people are the reasons we're going to win. Our product, as I've described it, very well designed for fit for use. Our people, our chief medical officer is the only person in the world who has developed 2, both of the only drugs approved for lupus in the United States in the last 65 years. There's a lot more I could say if I had more time, but the people are extraordinary, and the results in our clinical program are pretty clear. Our manufacturing strategy, this is the relevant question for our company right now. We're going to cross the finish line first to launch. The question is, how are we going to supply the market? And I will tell you that under my watch, we will get to launch the first product and the broadest portfolio, and I'm not going to let somebody else come in and take that opportunity away from us. So we haven't solved that yet, and rightfully investors should be asking, how are you going to solve that? We're on it. This is the group that's on it, been together for 5 years. Nobody leaves, nobody comes, just the same team doing a good job. I appreciate your